The brain is really interesting. At rest, it makes up about 2% of your body, but it's actually consuming 20% of the oxygen you breathe in, which is really what drives metabolism, a big driver of metabolism. Even at rest, your brain is consuming tons of energy. So metabolism is essential to everything that the brain does. So after a traumatic brain injury or an epileptic event or a stroke, you lose that metabolism, and what that ends up doing is actually either killing neurons or causing cognitive dysfunction at extreme rates, which is one reason we try to target metabolism, which I've been trying to do for 20 plus years, at the level of the mitochondria, which are really the powerhouse of all cells. Taking central nervous system metabolism, which is a focus of many research labs here at the University of Kentucky, as a theme for COBRE, was actually built way back in 2013 when Brett Smith, who was a professor here, eventually a chair of neuroscience, started the Epilepsy Center, which was really focused on metabolism. So anything we do in the COBRE, from whether it's pilots or putting junior investigators in place, is actually a competition. So we make a call for project proposals, and then we actually have professors that are experts in this field actually rank what are the best projects.
My focus at UK is traumatic brain injury and understanding what happens after traumatic brain injury that we can target, that we can understand the basic mechanisms of. And so we're specifically looking at metabolism in that mitochondrial centric way, which mitochondria, the powerhouse of the cell, is very important. And the other interesting thing we're doing is we're using a drug to target metabolism that we hope can restore mitochondrial function and metabolism in both neurons and blood vessels, and try to see how that can improve behavioral outcomes after traumatic brain injury.
So our laboratory focused on a particular protein that is called TDP-43. Clinical studies originally associated the pathology with this protein into the brain of dementia, such as frontotemporal dementia. However, lately, the last decade or so, has been shown that TDP-43 is a major pathological protein that impacts diseases such as Alzheimer's disease and a range of diseases within this dementia spectrum of diseases. So we discovered a protein called EI5A, which is a translation elongation factor that we showed, and we have published about this research, that this particular protein also impacts the accumulation of TDP-43 into mitochondria. Mitochondria is actually an energy organelle, and it's extremely important for how brain handles energy utilization.
We're going to be looking at biomarkers for conversion to cognitive impairment in Parkinson's disease. So this is actually a really important topic for me personally, and also I think for the Parkinson's community, because a good percentage of people, maybe 50 to 80% of people that have Parkinson's disease, can go on to develop cognitive impairment. In terms of metabolomics, I think this is an underexplored area in the field of cognitive impairment in Parkinson's disease. So people haven't looked carefully to see if they can identify a biomarker profile in patients that are going on to develop cognitive impairment. If we could do that, that would really help us in terms of treatment and intervention.